Hello and welcome to this 2020 Virtual Rosetta Workshop. I'm Cristina Elisa Martina, I'm a postdoc in Meiler Lab since 1 year, and today I'm going to talk to you about the Rosetta scoring function. So, first question, why we need a scoring function? I have a couple of examples, but there are much more depending on what you are studying. And the first one is, if we have two proteins that has the same sequence but show different conformation, which one is the best one? Like, which one is the more stable one? Conformation A or conformation B? Another example is if we insert mutation that can be a single one or multiple in the same fold, which one will fit best for that particular fold? So we need a scoring function in order to give a value to each protein and also rank the different structure. So the definition of scoring function, that can also be referred as energy function, is that the total energy is the sum of weighted energy terms that are calculated as a function of geometric degrees of freedom and chemical identities. Now, before starting more deep into the scoring function, I want to do a little description of the history in the Rosetta community. So if you find some older paper, you may find reference to score 12 as the scoring function. Then we had Talaris 13, Talaris 14, and nowadays we're using reference 15. So the Rosetta community is updating over time the scoring function in order to make it more accurate to describe the biological systems. So another thing I want to mention about the scoring function is that historically the total energy was given in Rosetta energy unit, REU, and this was an arbitrary number that was not being able to be compared with kilocal per mole, for example, that you can have by experimental data or in molecular dynamics. However, with reference 15, the more recent scoring function, this is now possible, so we can convert the Rosetta energy unit in kilocal per mole. Now, we have different kind of scoring function. In Rosetta, the two main you will find during the tutorial are the low resolution and the high resolution one. In the low resolution, that is also called centroid mode or coarse grain mode, we have the backbone of the amino acid that is explicit, so each atom is described as a single atom, while for the side chain we have a sphere or centroid that basically describe implicitly the size and the properties of the amino acid. The reason why we want to use a low resolution scoring function is that it is more simple and is faster. So the calculation time are much faster than not using a high resolution scoring function. This makes it optimal for a global search of conformational space, for example when you do ab initio folding or loop modeling. On the other hand, the high resolution score functions is necessary because it's more accurate, and it can be referenced as full atom or all atom score function. However, it's time consuming, so this is usually used for local search of conformational space, and you will see there are many application of Rosetta in which you first start with a low resolution scoring function and then you refine with higher resolution scoring function, and this is to find a good compromise between calculation time and accuracy. So if we go back to our example, the things you have to know within Rosetta is that the lower the score is, the better. So if we take conformation A, it has a total energy of 253 Rosetta energy units, while conformation B as minus 82 Rosetta energy unit, and between the two, the lower is the better, so conformation B is the more stable protein among the two. The question is why the energy is different, so what does it change? And for understanding this, we have to go back to our definition where we say that the total energy is the weighted sum of energy terms, and we have to describe what are the energy terms. So before entering in detail about energy terms, let me tell you that they can describe a single body, or they can be a relation between two bodies, for example, two atoms, and this can be an hydrogen bonds, or they can describe the whole body. And here there are examples for each of them. So one body, for example, the backbone is considered one body. For two bodies, we have Lennard-Jones that calculate the energy between two atoms based on the distance. And whole body, we have, for example, the radius of gyration. Now, this is a list of all the more common energy terms that are used. We will go through them one by one, but the first difference we can make is that some are physics-based, while others are knowledge-based. So physics-based is basically what the terms say is based on physics, so all the distance between atoms and energy or electric interaction or H-bond are included in the physics-based. The knowledge-based are basically extracted by statistics that we know about proteins, so we can analyze the whole PDB database, and we can extract information about preferred angles or other information. So let's start with the physics-based. We have the first three terms that describe the van der Waals interaction, so the hydrophobic interaction between atoms. And as you know, the red line is basically the Lennard-Jones energy that you should have studied in physics. And Rosetta used these three terms to describe the same. So as the distance between atoms goes smaller, so as the two atoms get closer together, the energy will increase and we will have repulsion between the two atoms. While if we start to go from a certain distance on, like we have a phase where there is the attractive energy, but then if we are too far between the two atoms, we will have no interaction. So Rosetta wants to describe exactly this, and we have different terms. So the repulsive term will follow this basically line in which we have the atom pair distance that is very small, and while the attractive force will follow this part of the graph where we basically get attraction. So the next term, the next three terms are relative to the solvation. In Rosetta, all the protein solvation is implicit. We can have explicit water molecule if they are important in binding, for example, with a ligand or with another protein. But for only the solvation water that is around the protein, we are using an implicit term that is described by these three energy terms. The next one is describing the electrostatics, so the energy between two charged atoms that are separated by a distance. And you should remember the Coulomb law, and Rosetta basically describe the same interaction energy with the term, the electric term. Then we have four terms that describe the hydrogen bonds. We can have basically H bonds be inside the backbone, and we have two kind of basically hydrogen bonds inside the backbone: the short range SR or the long range. Here I just noticed they are inverted. Anyway, LR is long range, and these usually describe the energy that you have between beta strands and sheets, because they are not close in the primary sequence. While the short range described mainly the alpha helices, because you have usually an hydrogen bond between the residues I and the residue I's plus 3. Then we have the interaction between the hydrogen bonds between backbone and side chains, and then we have a term for only the interaction that are within side chains only. So with these four terms we can describe all the hydrogen bonds that are formed in a protein, or also in a couple of proteins, like in an interacting protein. We have a term for the disulfide bridge, because as you know, the disulfide is a covalent bond, it's not a low energy interaction. And we need to understand the distances and the angles and so on. And we have a term that will describe basically the disulfide as a disulfide and not as two individual systems that are close in space. Then for the knowledge base, we have the first two that are basically related to the FMC angles. So for example, the Rama-Prepro is describing the Ramachandran plot. So as you may know, residues should fall in the red areas, and if they do, Rosetta will give a negative score. and this is what we want, as negative as possible. While if they fell in a more clear region, the energy that will be associated will be higher, and this will affect the stability of the protein. Then we have a term that is FADUN for the side chains. So also the angles, the dihedrons that we have in the side chain have preferred conformation that are higher or lower energy. So if we fell, for example, at 90, 180, and 270 here, we can have a better energy term, while if we are in this upper region, the energy will be affected much more. The next one is, the next three are special case of torsion. So for example, if we take the omega dihedra, that is the one referring to the peptide bonds, we know that it should be only in cis or trans, so 0 degree or 180 degree, and if this is the case, the energy that will be associated will be 0, while if we have something around 90 degree, we will have an associated energy of 80 that will affect the stability of the protein. So this we know is not physically possible. The same is valid for the prolines. Also the proline has a certain planarity that you should respect, 0 or 180, and the same is valid for the planarity of the tyrosine, so again 0 and 180. The last term that we have in the knowledge base is reference, and this is a special case that is used only in the design. So this parameter, if you are analyzing only the same sequence but with different conformation, will not be taken in account, but if you start to design, these energy terms will take in account the unfolding energy of the amino acid that is known by experimental data, and will also adjust the amino acid frequencies during the protein design. So the next topic we need to discuss are the weights. We describe the energy terms, but to each energy terms is associated a weight that will give more or less importance in the calculation of the total energy. So you can see here in this yellow column the weights that are used for basically all the physics-based terms. It is 1 because we are reflecting physics data, while for the knowledge base, this term has to be weighted based on experimental validation, and so they are not necessarily equal to 1. So some of the weight for specific energy terms are set to 0, and so the term will not be included in the calculation of the total energy, but if you change the weight to something different from 0, you can include it. And another things that you might know is that if you need, you can modify the weights, and it was done in special application, but we strongly suggest you to not do it unless you are really aware of what you're doing. Anyway, if you need to modify the weight, you can use different methods. You can use the command line, and you can use the option score weights, and you add a file. You can set score, set weight, and here you put the name of the energy term that you want to change, and here the number of the weight that you want to change, and you can do for multiple terms. You can use a patch file, so this is an example of a patch file, or you can also use an XML script. And in an XML script, this is the, highlighted in yellow is the basically the command for changing the weights, and you put here the name of the energy terms you want to change, and here the weight that you want to change. So if we go back to the total energy and to our true conformation, we can see that, let's analyze the score file. And this is the score file. I guess you already saw some of them. It's really messy, but one thing we can do is divide by each line. So in the first one we just have the header with all the name of the descriptor for the columns. Then we have in red the description for the conformation A protein, and in the green we have the conformation B. So in the second column you always find the total score. That was 253 for conformation A and minus 82 for conformation B. But you can also check in all the other column the energy terms that I just described, so the attractive, the repulsive, the solvation, and so on. So if we go to see our two conformers, we can see that they differ in the total score, but they differ also in the attractive and repulsive energy terms. So B is more negative here and less positive here. So what changed between the two is that the Lennard-Jones interaction are improved in conformation B compared to conformation A. And if we change the view of this protein, so this is a top view, and in red I highlight the hydrophobic amino acid, you can see that this conformation is less packed, so all the hydrophobic are exposed to the solvent, while conformation B is well packed, and I think their balance are interacting between them. So this is one of the reasons why conformation B is a better model compared to conformation A. And understanding each energy term will definitely help you to understand what is the biological problem in your structure. So the next topic I want to discuss with you is the Monte Carlo sampling in Rosetta, because this is really important for the conformational landscape. So let's say that you have a first model that has an associated energy up here. Then you do a second model that has a lower energy. That is what Rosetta wants. So the Monte Carlo sampling will calculate that the new energy is lower than the old one, so we'll accept the structure. But let's say that then we add a third model that has higher energy compared to the old one, that is the blue one. The Monte Carlo sampling will at this point pick a random number between 0 and 1, and if the difference of energy calculated here is higher than the random number, the Rosetta Monte Carlo sampling will accept the structure. If not, it will discard the structure. This is very important because, as you can see here, the blue protein fell in a local minimum. That is not the global minimum where we expect to find the native protein. So in molecular dynamics, for example, it's really hard once you are in a local minimum to go out and pass the energy barrier. But this Monte Carlo sampling will allow you to sample all around and pass energy barrier so that you can finally find the native structure that is in the global minimum. So other things to mention regarding the scoring function. I described low resolution and high resolution, but there are special cases like membrane proteins. So as you may know, membrane proteins have two parts that are exposed to the solvent, so the extracellular and the intracellular loops, for example, but there is a whole part that is inserted in the membrane. And the membrane itself is not just hydrophobic, there is one layer that is polar, then there is an interface one, and then we have the hydrophobic core that is really the central part, and then as we go up we find again the interface and the polar portion. So we have to score differently this portion of the protein that is inserted in the membrane, not only from the soluble part, but also between, like, we have to make a difference between this polar portion and the hydrophobic portion. So there are scoring functions that are dedicated to membrane protein only, and if you are working with membrane protein, you should use one of these. Now, we have people in the lab that work with membrane protein. If this is your case, be free to ask them on Discord or on the chat. Then other things that we want to use and we want to implement in the score function is the constraints. So usually we have experimental data that tell us, for example, we know these two atoms are at this exact distance, and we want to include this information in the calculation that Rosetta does. So what we can do is using these constraints that are sometimes referred as restraints, between, for example, atom 1 and atom 2, we want 6 Armstrong of distance, and if the Monte Carlo sampling falls outside this 6 Armstrong of distance, the model will be discarded. And these help us to basically include the experimental data and also to enrich our design with good models. Now, The next topic is biomolecules. So we can insert, there are energy terms for non-canonical amino acid, in case you need to work with them. There are energy terms for carbohydrates, like glycans, for example. There are energy terms for both DNA and RNA. So all these are set to 0. The weight is set to 0 because they are not used always. But if you need to use it, you can just set, like call them, and they will be activated in your system. The last thing that Rosetta can do is integrating experimental data, for example, for electron density from residual dipolar coupling or for Sachs, into the score function. So there are groups that are working in many different experimental interfaces with the computational side, and many of them, I think you will have the electron density tutorial later on. So that is all for the scoring function. You can find more information in the paper for reference 15 or for older scoring function. And I'm sorry, the link here is really clear, but you can go to RosettaCommons and see the score types and the explanation of the scoring function. Don't hesitate to contact us if you need something or you have other questions. Be free to contact us on Discord. So that's all for this tutorial, and see you later.